Thank you. Okay, guys, we're back online. We had some connectivity issues to solve, but so we are moving for the next station. I hope that everybody on storm watch alert are okay. The weather here in the Gulf of California is beautiful, though, but just keep safe. Y todos los participantes y eso están en el de Falkor. ¿Y son los mismos? ¿Eh? Sí, están haciendo 50. Espérate, yo te digo. Oh, wow. You good? Okay. Ready. ¿No te sabes la dirección de internet? No del siglo del SuBastian, pero del Falkor, para ver el participante y todo eso. Sí, según yo.
Okay, let's get the probe in, please.
I'm just gonna lift the business just a little bit over here. 2 seconds here.
Pero no me deja. Está aquí participant data, pero.
Alright, I think it's pretty soft there. Ready?
Let's go ahead.
Okay, coming
Ah,
down.
aquí hay otro. Creo que es esta, ¿no?
Stand by. Okay, you ready? Sure.
Ah, nice. Super soft.
All 20 pounds of them, or just 5? How much you pay for them? 40 bucks. Yeah. Kilo and a half. No, for my niece. Yeah.
¿Y dónde están las fotos de Flores? De Mónica.
Ah, todavía no los he subido.
Pero no las has encontrado aquí.
Los de nosotros, como yo le pedí que los suba ya.
Ah,
Nosotros, de nosotros, pues,
sí, el único que ha subido. ¿Fotos? Sí, si nos preguntabas que las ibas a subir las fotos y los videos.
Voy a subir, no me dan chance de ponerme a separar las que son de este leg y del anterior, entonces lo que voy a hacer es subir todos los selects, todas las fotos que he seleccionado de ambos legs.
Y no te olvides de enviarme los videos.
Los videos también los voy a... Siempre, yo estoy medio muerta, siempre. No sé cómo es, bueno, es que tú estás toda congelada.
Sí, y además mi agua siempre está calientita.
O sea, me destruiste, quiero que sepas.
Ay,
Luego
no
tenemos
manches.
que hacer yoga de cadera.
Sí.
Ah,
¿Te sirve que seamos paintballs? Que me duele exactamente ahí abajo.
vamos a hacer llamadas así de ¿cómo se dice? Terapia, de support group, sí. Y esto te alivia, y después de estártelo...
Sí, no, y hay uno que es como una pistolita.
Ah, que hace pa.
Y aquí
Sí, exacto.
Exacto. Y se te puede venir así, todo.
Sí, porque siempre estaba como localizado donde estás ahorita, pero después de nuestro ejercicio se movió para acá. O sea que sí le dimos le digo, espera que todavía me duele, y él le dice, o sea, ni siquiera hicimos ejercicio.
No,
o sea,
nada más
tiré por 4 segundos, estamos de acuerdo. Y si te duele, me dijo, ¿good sore o bad sore? Y yo, espero que bueno, porque significa que le movimos. Justo vine, muchas gracias. Justo vine a tomar fotos porque me di cuenta, estaba seleccionando y me di cuenta que en este leg me he enfocado mucho más en video que en fotos. Casi no he tomado fotos, entonces es como última oportunidad. Perfecto.
Bueno, no se te olvide ponerme.
No, para nada.
These kids are even.
Yeah, the ones that have Ethernet, one of them says like,
Right.
just like, let's see.
fotos que está
No, en general las que
Ay, sí, ahorita es lo que me di cuenta que me ha enfocado mucho más en video que en fotos. Este LED, tengo videos.
Okay, let's get the probe out, please.
Probe coming up.
How far are we going?
Give me a second.
Yeah. So we just have a little connectivity issue. This computer locked up.
Entonces ahora está 50.
Keep back now.
Yo creo que sí, ¿no?
Yeah, just probe holes in
¿Cuánto
there.
crees?
Cool. 50.
No, pero ¿cuánto crees que va a ser esta?
Está por encima, todos se ven parecidos.
¿O 6? ¿Por ahí?
Sí, yo sé, pues,
Ok. Entonces 50. I think it's gonna be another 15
That's
meters.
gonna put us really close, 10 meters away from this heat flow that we did. When we were going from up here.
Oh, we're already there?
Yeah.
Okay, then make one
And then,
60
then,
meters away from
then,
there.
then make it 50, 25, right?
Between,
Yeah.
Okay, so
between.
20, right
Yeah.
back. You want it in between or 25?
In between, please.
Roger that. On the 10.
So how long for the in between point? How far away?
It's gonna be 30 meters away.
30 minutes. Perfect.
Well, aside from the crash of the computer, but it only cost about $10. Well, it was a complete crash. They're supposed to be like this computer integrate runs the whole we were preparing a food. No, they kept the science of the world. Yeah, so there's some moments of terror. And then
¿Se movió?
that's why you had something.
Siempre pasa.
Do we need to think about the next scope for one or no?
The next first?
Yeah.
Yeah, yeah, but when we get into an area of good hydrothermal mat and plants, is,
It's when we
Yeah.
want to do that.
yeah, so we've already taken 6, but I talked to him to get to 7,
Right. No,
but we haven't gotten to the higher.
no.
The 6, right?
Yeah, I mean, we almost got stuck with the first part of the transit that we did the other day. So, I think we're going to start going a little bit north to fill some gaps that we have.
Yeah.
And so I don't know if we're going to really get any closer to the vent.
He, he doesn't need that core, I gotta talk to him to do it. So we don't do it, I think it'll be fine, but we're gonna fly over some of that stuff, so we're gonna want to keep close station next to it. Real quick to get one.
You mean because we're gonna fly over that patch?
Well, there are a couple batches on our track, not only at the northern end, but just north of the second one, there's some bats up there too. see one and you're gonna push core, I mean, we're doing heat flow in the area anyway, then I would do.
Well, we're about to get to a point where we would,
Leave
in the discussion yesterday, we would jump like 300 meters or something to get
So
in
you fly off
between
one of them.
those two measurements where it dropped it off.
Yeah. So you'd fly off bottom then.
Yeah.
You know, he wasn't really even expecting this, I just told him I thought it'd be nice to have, so I wouldn't take a long time again. It's my opinion.
But I think we're doing good.
Yeah, it's been very interesting.
Good.
I really think now we've got segmentation, but along a structural control that's that extends through the whole southern part of the basin.
Yeah.
And then the different
We're landed.
cells. Now, the question would
Thank
be
you.
whether it's different cells of upwelling or just only limited places where you get out.
Where can you get out of this?
Yeah.
It's not bad.
So it can be controlled by permeability or flow characteristics.
The spacing of these big mounds is not dissimilar from the spacing of the three big
Can you
mounds.
increase them out a little bit?
They're not exactly the same, but they're kind of on the same scale.
Yeah, they are. Yeah, it's a little further apart, but you know, it's certainly, yeah, it's more than 50 and less than 200.
Yeah. Let's see how this will change.
You want me to, like, over here, or?
Yeah, let's find out if the other line.
Okay.
Yeah.
Okay. Yeah. Wave that again. So Monica gets a picture.
We're already
Thank
past,
you.
okay, we're already past the central line.
Yes,
I didn't know we'd already had
Yeah,
that.
it was for real.
So the next thing we do is leapfrog all those items.
Era broma. I have to be switching between video and photo all the time, so it's difficult.
I'm always joking around, you know, something
Let's
funny
insert
happens,
the
I
probe,
didn't
please.
get it, and I said, hey, do that again.
Get the probe in, please.
Video.
Yes.
Probe,
Here,
yeah.
we need
Probe
this.
in, please.
In my first expeditions, I would be too shy to ask a scientist to do something again because also I thought they are not here to post.
Probe's up.
Now
Thank you.
I'm like, I'm sorry, but and especially if I know very much stress.
It's gonna be a complete one.
Tell you're a little more used to us putting the bosses around than you were the last time we were out.
Hopefully in a nice way.
Oh, of course. You always said it.
Yeah. No, the first time I came was 2015 and I was so completely
Bigger spacing. What the blue dot is.
now I'm not willing.
Oh, okay, yeah, that's
That's
good.
that is our first filling the gap point.
Okay, well, there's gonna be clams there.
Oh, okay.
They're not,
Yeah,
that's
we-
not the densest spot, but there's gonna be clams
Yeah,
there.
we are starting to getting into this
Yeah.
high heat flow.
So when we land, we should reconsider whether or not to do a push point. Yeah, that's good. That's an area where we want to be doing.
See, just summarizing, so we've taken push cores from distal, two from distal parts.
Yep.
Okay, and then yesterday, one from Diane Spent and one from Marathon. That would be the hot,
Yeah, Diane found the matter artist not quite as low a temperature as
Right.
I thought it was, but it's still low.
Yeah, so
it
that-
actually does have different mineralogy, and we'll see if it has some chemistry we get back there. I'm convinced that mineralogy. So I think it's really worth comparing those two.
okay.
Yeah.
And then
And
what
then we
about?
took that one core that was next to a little chimney that we were gonna sample, but then we moved it to the sampling, and it's not gonna be very useful. But, you know, we already took it, it's already in process, so,
Right.
but that was definitely inactive,
Right.
well, inactive deposit, but we have a chimney that's inactive. But anyway, we know the content.
Okay.
Okay, so what we don't have is something that's sort of a moderately active, pure land.
Okay, so that's what we're looking at.
Just do that every day. I send it to Chris. I don't know.
¿No te acuerdas si esa última medición hicimos cerca de aquí tenía conductividad?
Los últimos dos no.
¿No? Entonces hay que hacer esa...
Sí.
Have email. Looking at my house, apparently it's when you get them.
Yeah, it is. I haven't heard anything wrong with it.
That hasn't happened in a long time.
Yeah, there's something about, there's one town that's expected to wash away
Already.
Davis or something
Yeah.
like that.
¿Y los datos en qué folder están, Chloe?
It's flat.
Ajá.
No, los puse en
Washes away half the
Todos los
state's
otros
gonna
todavía.
Ok.
Well, you know, historically that's happened.
Oh, well, historically, Davis was a state that's swamp.
Much more productive biologically.
Yes.
Yeah, I don't know. Last year they actually evacuated us for the rain and everyone had the expectation that, because it had burned everywhere around where we lived. And, there were some flash floods down the road. But nothing grew because that was the only rain we got. So nothing grew last year. Yeah, so I'm not sure it's really a different story this year. The mountainside above us is still basically bare.
Pero también está en un lugar muy caliente y todo bastante dulce. El tema de pues prefiero casar con dos, sigamos el plan. O sea, antes.
That's working. Those guys. It's really a huge effort to manage that. They just hold dial no, it's the science part. I can't remember what just have a requirement, set of requirements that they really never, that it just happens. Well, not knowing anything cash flows. The scary thing for me is that if we get this on Russia, all of a sudden, for the very first time, I'm going to be the responsible person for a shipboard and I'll take it to I'm so close to being but I've never been in a position to have it. Yeah. Joke about making genuine. It's not, that's not where it's going to be. Yeah. She's been following along. Oh yeah, no, she's really involved in this group, even though she's not here. Oh yeah, I know. The thing that's going to be hard about that summer ship is operations wants to have nothing to do with it. I think it's just kind of crazy, but yeah. They do, right? They don't like to take on responsibility for anything in addition to their stuff. Yeah. It's easy for me to say, I'll handle the data. Yeah. But I'm absolutely not capable of handling the electronics or the interface and things like that. So, well, it doesn't get run except like that. That's how it works. The way I've already runs our ships, you go to the same place all the time. Right. It is a system that's justified on the basis of data has to go on expeditions to someplace new, map it, A/B it, ROV it. So that's all it wants to go places and have things. Well, sure. But he takes you with him. But that's 1/10 of the activity of the ship. More than 50% of the activity of the ship is driving the same track line at the same points in Monterey Bay. It's going to get turned on every few months. Another reason why it's okay that it's not a great installation. Because it has to be a lot more. But at some point, operations will come to recognize that somebody with an electrical engineering background needs to be involved in this. It's either go out to sea. Well, I mean, you can't because you have to make it. That's either they seriously don't want that to be very
Sí, eso es justo lo que estaba pensando. Como este leg fue tan corto, hice el video de la así y
that's
no
a
hay
third
un
order.
momento en el planeta en el cual va a estar esto en el LED mañana, antes de que se baje, pero sí va a haber, de hecho hoy iba a empezar a trabajar en eso, pero luego me acordé que casi no he tomado fotos, entonces me entré en pánico y vine a tomar fotos. Y justo estaba pensando eso porque tú no vas a estar y Dave no van a estar, ¿verdad? Entonces lo único que van a estar, porque necesito que uno de los científicos principales lo vea para probarlo antes de mandarlo, entonces supongo que va a ser así, pero como mínimo quiero ver si entre hoy y mañana alcanzo a editar las entrevistas, como sería súper aburrido para ti porque estarías viendo el video, pero como para que oigas cuáles son los extractos de cada entrevista que voy a usar y me digas falta esto o esto no es exacto, ¿sabes? Como que me das tus impresiones, pero no va a estar ni la música ni falta solo bla 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 bla, pero
No, pero sí, haces muy buenos trabajos. Así que yo
o sea, de que obviamente va a haber un video de este Lexi y sería, pero porque tú corriste ayer, yo y me muevo y todavía me duele, no me jodas.
Le pedí a que la pusieran más.
Hablen despacio.
Estos son los pushkits que vamos hasta ahora de ROM. Entonces son 3 en JaichMaa y 2 en ROV, pero no sé si aquí va a
Okay, get the probe out, please. Rope's coming out. Thank you.
Entonces, ahorita espero que vayamos a tomar un poco. Y ya sería el esfuerzo que sería el último.
Sí, serían 6. Y bueno, el 18 es el que está en Isla Cerrado, que se nos sale.
Rope is out. Thanks.
Y ahorita te voy a dar.
Ready to move?
Sí, please.
Modern constitution.
Te voy a dar las coordenadas del discurso. Sale. Pero está súper bien. So sí, so my net carrying out work on this figure. So okay, one. And so that's so far this is our path into the and then once here
Yeah, so the firm right now.
yeah. And then this was also the background.
That's the little I connected to.
Oh, okay. Okay. So we definitely, yeah, if we can get something here, yeah.
Yeah, I'll be in there and watching it. It's 600 station. Tell you what.
Yeah, the 18 one is from Surround, so that was
Yeah, okay, I thought we had already taken 6.
Oh, we took 6, but it's one of the washed.
Oh, that's right.
Yeah,
Yeah,
remember, one was,
That's what it is.
Passing over a clam field,
That's why it was good. Got it.
go see the clams?
yeah.
Yep.
Go to the clams.
so the plans coming, that probably went to God. All right, thank you.
But don't core clam,
No,
because they get really sticky.
Right, you can't throw a sample away.
You know, actually, it's kind of hard not to sometimes. But for every clam you see on the surface, there may be 10 other surface medical clams.
I don't know
Right.
if it gets older than the shelter.
hopefully not. It's not going to be the case.
See, we're gonna climb back out of the bed though,
If you look this way, you can see the time on the edge.
maybe unless we get there.
That really be closer to back to her.
So we're aiming to take 2 measurements close together here because we have a big jump from 6 to 30. So we're gonna already point to the one that is big kind of on the middle, but I'm gonna be in close space, and then a second one because it's a big jump, and
Yeah.
so we wanna look at it with high resolution. And after that, can you put the map together so we can explain
No,
the
4.
so
Yeah, and on.
that.
Yes, yeah, right.
So this is up here.
We got at least 4 stations.
Yeah, no, it's.
So that's already 2, right?
Yeah.
And then after that, we could do what I would definitely 1 here,
Right.
between the 2 and the 7, and
already accomplished the long.
the long transect. And
Yeah,
really the
yeah,
only
so
difficult would
I
be
think this
that
is
one.
good.
Yeah. But that's, if we don't have time to do it, it would be as difficult as.
Yeah, right,
So,
yeah,
okay.
that all sounds great.
Okay. So.
Okay.
Okay. So we're ready there? So there you go. They're coming where it's hot, and the clams start to show up. So this is a beautiful clam field, and then J-Rod is ready to poke. So we're gonna go into the sediment and hopefully not disturb any of the clams that are down there. It seems pretty soft to me, so
It's soft, but it's a little denser than,
okay,
but it's still, you can get the
cool. Nice. So are you ready, Flo? Sorry. It's soft, huh? Nice. So, well, I don't know. See, you think this is? We should wait until the two
It'll
that.
be an okay place for a quarter, but we have like 2 more chances.
Yes.
So do we wait or we do it?
Okay,
Did-
get the pro in, please.
road going in.
Yeah.
I'd like to see a bacterial map, and I think we'll get something as good as this in one of those 2 places. Let's wait.
Let's wait. Okay.
Let's wait.
You got a whole nother leg to pull off those.
Hmm?
You got a whole nother leg. Get those fish doors.
Are there any Solomaya out there?
Any what?
Solomaya, that's the clam, I couldn't remember the name.
Oh, okay. Selma Maya.
Solomaya.
Solma Maya.
Solamaya.
Solamaya.
Yeah, and I didn't remember, I had to go back to my old dive notes, look it up, but I found it.
Is that related to a Vescamyid, or is it a different type of clam that's also
They seem to me to
chemosynthetic?
be, they certainly live the same lifestyle and they're kind of the same size and shape. But I don't know what that means in terms of what biologists would say about how closely they're related. They are sort of different families, if you will. I don't know if it's really family level, I think it's family level. Yeah, see, all of these have sort of a dark skin on them. But when they die,
Yeah.
the dark skin goes first. And they don't.
Okay.
The other ones have almost like an armor coating that, but after they die, that will come off. And it'll persist. It'll
Hi,
stick
fella.
around for a long time, even after the whole clamshell's gone.
You know, we're not seeing a lot of Xenotribella in these clam fields.
No, because we're kind of in a dead field.
Is that a shrimp, Shri?
Is it a what?
A shrimp?
Oh yeah, that's a shrimp. You want to take him home for the barbie?
May or may not taste so good. That's
Well,
what
we
they
had
say.
a good shrimp last night.
Yeah, we did. I don't think they came from hydrothermal fields. Yeah, shrimp is great, but probably doesn't go well with hydrogen sulfide.
He's a scavenger. He's not a vent shrimp. There are vent specific shrimp. Like Rhymacharis ex oculata. Russ, and I know these things.
I was just thinking to myself, Z is showing off again, man. Z is showing off.
That is, and Rhymacharis ex oculata is the favorite food of the famous vent kitty.
The famous what?
Vent kitty.
Vent kitty.
Yeah,
Okay,
vent
explain,
kitties.
please, Z.
I'll have to pull up an old file and show you. But we have pictures of vent kitties
Okay.
from Escanaba. I'm sure I've got that somewhere on the computer.
Yeah.
How far do you think the road is? Are you gonna wanna fill this gap right here? Between 30 and 31?
Between
32 and 48?
32 and 48.
Okay, so right here.
Right.
Okay.
Then we will definitely want to go. Yes.
You want two to go in between there, or just one to go there?
I think.
Let's see how the seaflow changes.
Yeah.
And maybe decide on how the clamps change,
But
and
yeah,
maybe we
here, between these two things.
get, we get another hour, so 3 more, then that's it.
According to the map, we'll just take it. We'll just take the dip.
Uh-huh. Oh, yeah, I see. Oh, that's right. Yeah, so they were just pointing at me. I didn't see that 3D bathymetry map. So cool. So please note that we are like in a depression right now. And that's something that we're, at this scale, looking at the ROV cameras, we could never ever see that. But that's cool. And we will continue on this path north. So let's see how the sediments change and the density of life changes. That map that we just showed is looking up north or south?
North.
North, right? Yeah.
Success. Okay, get the probe up, please.
So, coming out.
Thank you.
Probes out.
Thank you.
Thank you.
You didn't leave any of them out. No, I don't believe so. I don't believe any of them escaped.
That's good.
Go.
Okay, ready to go?
Yep.
Boogity, boogity, boogity.
this. Show up
Let's
a
not
few
talk
times.
over me. Did you see that? Did you see the email exchange?
No.
No, I couldn't. That was
No.
so that was
Okay. Or later.
Spider. That looks like that.
Yeah, let's do just one before the field comes up. And then a few more meters maybe. Right? And
A few more meters forward.
then maybe a meter or so. Okay. Maybe.
What, you wonder how far away that is?
Where's this field?
Must be that only pressure right there.
It's about 3 meters this way.
Think so?
Yeah, that's 5.8 to the line.
Just stop right here.
Oh, if it's that, then yeah, you're about 8 meters.
So just keep moving forward. You want to get right before?
Yeah.
Okay.
Meter more just before the field starts. Okay.
Great.
Okay, so we were transiting, thinking about going around in the middle of 2 points that we already taken, but we found this matte concentration of a clamp field, and so we're gonna
Boom.
take a measurement right before we enter the clamp field here.
Or do you Sure.
Yeah.
I asked first. But you weren't listening.
So as we zoom in, you can see that this is clearly a clamp field, and I think we will have plenty of chances to see different anemones and the fish.
Safe bet.
Ah,
Okay.
this is, please. Okay, that was perfect. Thank you.
So how far are we away from the last measurement we did?
The last one was 48. You measure it pretty fast.
We are approximately
14. Okay. What do you think here?
Is this, did we move?
Yeah, we move only 14 meters.
Yes. See, there's some more flow out here. There's more bacterial.
Yeah.
If whoever wants to focus on there, we see that. I like this better, but plan for your because you do it. So.
Yeah. Well, we're just on the boundary.
Okay.
We could not necessarily have to hit one, I guess.
Yeah, I think in front
Yeah,
of the
no,
software, we
that-
could probably get away with it. Yeah,
Okay.
I say yes.
Okay, so let's go for it.
Yeah.
Voy a hacer un push core aquí. Okay. So, okay, when we're done here, we're gonna go for a push core.
Okay. In this location?
Yes.
And please don't get us new plans.
Yeah, only get plans that aren't gonna smell bad.
You wanna play with the camera for a bit?
Oh, I thought we were going to get Oops. It's okay if I zoom.
Oh, of course,
Like-
all yours.
Okay. Yeah, it looks pretty good for core over here. Looks pretty good for a core over here.
Zoom out real quick. Where you're at?
Oh, yeah. Sorry about that. Oops. Dyslexic zoomer.
Okay.
Yeah.
Okay, go ahead.
In the best place, the easiest place to core is the best place to core.
Two plans or less, right? 2 plans or less?
That's right.
Did we get plans in level?
And that scale worm is an orphani.
You say it's an orphan?
There's another nice purple scale worm. A little bit of bacterial mat. And there's a squat lobster and Galatheid crab. Oops, sorry about that. Oh, who's the orange guy here? Can we get him in focus? J-Rod, do you know who that is?
I do not.
I
Look like one of those worms that flip their hair back and forth. But
okay. Yeah, I don't know who that was. If anybody wants to chime in on that orange wormy-like critter that was in the water column, we'll try and pass it along. I don't recognize that thing. But then I'm a geochemist. I know those are clams, and that's about it. I'm pretty sure they're vesicomyid clams. I know they're chemosynthetic clams.
is that a can or a bottle? Do you know that?
No. Where?
On right of your screen?
Yeah, see, on this. No. Here.
No. Oh.
Here.
Oh, I see. Yeah.
That's not natural.
And I'm not sure what that is, but that is not an animal. It was dropped here by an animal, and not very considerate animal. It's been here for a while. I'm not sure what. Looks like a bottle, I think. The little pinky tubes, like, oops, I am really dyslexic today. Sorry about that. Okay, that little tube that's facing away from us, that's one of the clam siphons. So there are a lot more live clams buried in the sediment. And you can see the little siphons sticking up where they bring oxygenated seawater down to feed their symbiotic bacteria. And their foot is in the sediment. And the sediment is more reducing, and they're probably getting some little hydrogen sulfide across the foot membrane and feeding that to the bacteria. So the H2S is oxidized by the bacteria, which are symbiotic in the clams, with the oxygen. And I'm looking in the middle here. If Greg Rouse or someone else is out there looking, is that the black thing, is that from a Solomaya clam? I have seen some of the dark skins from Solomaya clams out in here, but they're not very common that I've seen. So maybe somebody could chime in on that. Lots of anemones. And look at just the density of the clam siphons. Now you can see some of the clams. If I remember right, there are at least 3 species of clam here, including a small one that you don't see very often. It's in the sediment, but I think maybe we're seeing some of those at the surface here. I'm not sure. And we're pulling out the heat flow probe. And we're going to do a quick push core here that will be used for geochemistry. We're going to do the push core near but not in the clams. We really do not want a clam in the push core. They don't smell very well, very good after a few days. So. And so Jason in Michigan is suggesting that the orange thing may have been an arrowworm, but thinks that not so sure because of a weird color. I'm going to take his word for it because I don't know. So we're going to pick up core 2B. Bae?
Mm-hmm. Mm-hmm. Thank you.
Yeah, please. Whatever they are.
Well, as it zooms in, there's just no light stops.
Say, only certain.
Oh, there's
There might be a claim in there.
There's definitely a shell in there. You should note, actually, in the notes, that there's a clam shell about a third of the way down the core. You could just do another one.
Oh, yeah, I brought a note.
And so when you guys split that, be worried. I'll go warm it up.
Is it?
Yes, very happy. Thank you. So now, the idea is to fly a big mass.
Okay.
She was all over. She already had it dialed in.
¿Cuál es la otra? Pero dice que ya no vamos a quería explicarles justamente.
Yeah, that's what I mean.
So
Did she get here?
he has to go like
That's
somewhere in between these two guys, and also to correlate with whatever is
The other
happening
day,
here.
I was
So
talking
we reach like the
about
other end of the material map, so that would be a good point.
and then the side.
Yeah.
Do you wanna
So,
Go around it and just look at it?
well, yeah, I think just, well, fly around it.
Yeah, that way we were looking at it, so you know how far away we are.
Okay.
Like, we
It's part of the reality, so I'm not quite used to
can
it.
go by distance, we can go by sight.
I know, but I don't wanna go too far away from this line. So I will just go straight and let's how we see things from there.
Okay.
It might not be as clear as a boundary as like right now,
Yeah.
right? Like bigger. Yeah, so it's like that's what the next it's too close to make more. I probably should just go ahead and try to be in between these two lines.
So like right here?
Yeah.
Okay.
Está muy chiquito, bro.
Okay.
Okay, guys, so after that amazing clam field, we're just moving on. It's probably be just a few meters after we stop and fill a gap in between a couple of points that were especially high in heat flow value. So we're just gonna wanna see how it correlates with density of clams and biology that we see around here. So how far away are we from the last one then? Sure. Thank you. Sediment, ¿no? Para la siguiente.
31.
31? Okay, perfect. 31 metros de la otra. So, ¿ya están listos para...?
Sí, esto de las terapias, sí.
Ah, ok. Explore. You want to take a look at what you find here? It's not as exciting as where.
Go ahead. No, you're on it, man. Go ahead.
Okay, let's get the program, please. We're all going in. Okay.
Go back down to the little stock guy. Look like you have a little clean on the side. It does. Or is that just a worm on it?
About this?
Yeah. Oh, that's him. Yeah. He's got little purple tentacles.
That's better focus for you right
Yeah, there's a little stock creature that there's a purple thing on the top. I'm not sure if that's part of the stock creature or something that's just colonized it. Don't recognize what that is. If somebody knows, feel free to chime in. It certainly just could be an anemone on the stalk of something else that may or may not still be alive.
sediment's always amazing.
As Lisa is noting, we don't see a lot of stocked animals down here, and mostly umbrellas. I've worked at another sediment-covered area that has hydrothermal vents. This is on the Escanaba Trough. It's about 3,300 meters of water, so kind of similar depth. And up there, we've got a lot more stocked animals. I'm not sure what the difference would be here, but there really aren't a lot of sea whips or sea pens or gorgonians or things like that in this area, even though they're kind of similar depth and, to me, similar type of environments. But maybe an ecologist out there knows why that might be. This is a much more productive area in terms of surface productivity compared to the Escanaba Trough, but since the stock things are filter feeders, I think maybe they would be more abundant here, but that doesn't seem to be the case. I guess he wants to be in the picture after all. Oh, so now it's a Zoarcid party. There's one of those little red benthic jellies that has been named earlier in this cruise, but my mind being a sieve, I've forgotten who that is. But we've seen him before. He's got to be the center of attention, doesn't he? Oops. All right, we'll put you in the middle. You could tell he was unhappy when he wasn't in the center of the picture. That's why he's pouting. So our bottom temperature here is about 2 degrees Celsius. Yeah, 2 degrees. which is pretty typical when you get down to the deep oceans. Temperature is usually in the order of 1.5 to 3 degrees when you're below 2,000 or 3,000 meters.
So,
That's what I was talking to you guys about this morning, is that we multi-dimensional design with very precise locations.
thank you.
Oh, I was looking at it. Thank you.
All right, so what's up?
That's a benthic jelly. It's been seen and named on the chat several times, earlier times, but I've forgotten what it is. But
Okay,
but
there's
this
a bunch of these already.
yeah, well, not a bunch. They're not. We've seen them, but why they sit on the bottom, I don't know. Maybe somebody out there would like to tell me.
It relates to food somehow. So I don't know if you saw what I put in here.
Nope.
that's 25 centimeter contours, 400 meters across. You know where we are. The reason I showed it was when I look at this, it looks more like an erosive channel than necessarily a closed depression, which is what I was talking about before. And so it might be that the clams are eroding that out, but it also might be that erosion is unroofing the superficial sediment, exposing more enriched, deeper sediments, and that might have something to do with the...
If there's any kind of tidal flush, you know, you're going to get these eddy currents around an edifice like that, but that could be the case there.
Yeah. So
Interesting.
So it's another possible mechanism.
Yeah.
On top of everything else we're thinking about.
Yeah. Oh, that's cool.
Okay.
So we've got about 20 minutes left before the end of this dive. We're going to try and get another 1 or 2 heat flow stations in. We're taking the probe out and we're going to get ready to move up to the next one.
Probe's out.
Thank you.
Yeah, maybe a little bit closer. Yeah. Like,
Just come,
just a bit, yeah, just a little bit closer, just right on. Yeah, that's perfect.
looks good.
Yeah, that's it. Okay. Perfect. 92 metros.
Y aquí.
Sí.
All right, moving now.
Okay, perfect. Thank you.
No, I haven't heard this one. Under the impression it's Sunday.
Really, do you have any position? Yeah, roger that. Moving now.
¿Por qué es que estamos rellenando las cosas?
You like the glance? Maybe someday you'll get a chance to go back there.
Oh, wait, bridge. Yeah, I'm just gonna move my heading to make that transit.
Sorry, I messed with you.
No, that's fine. No worries.
Roger that.
to start another measurement in here. Have a clear so far. It's fine.
It's okay, we can live with that. So we don't have to stretch the time and get everybody tense. Especially because, well, we don't necessarily have that one to be complete, but I'm not sure. I think we'll be just right on time for this one.
What's that?
I think we will just be right on time for finishing this one.
All right.
I have to write a blog,
Oh, yeah.
and I feel so uninspired.
I've had trouble getting
There's a question on there, guys.
So there's a question. Do you want me
Oh.
to answer that, or do you?
Oh,
want
well,
me to
the about Victoria's Lake?
Yeah.
Or what?
Yeah, the focus of the
Yeah,
biology.
go ahead.
like-
Go ahead. Do it better job.
So on the chat, there's a question of what's really the focus of the work on the next leg. This is Dave Caress jumping in. And so there's a lot. There's a lot that's going on in the work that'll happen on the next leg, which is going to last a full 2 weeks. Legs led by microbiologist from Caltech, Victoria Orphan, and also Shauna Gofrady, a marine biologist that specializes in organisms that have chemosynthetic symbiosis, and she's from Occidental College, and then Greg Rouse from the Scripps Institution of Oceanography, who is a pretty general marine biologist with a real focus on worms and other invertebrates, and together they're looking at the life that inhabits the seafloor and the water around these hydrothermal vents at a variety of scales and from a variety of perspectives. And so they are looking at the very zeroth order question of just what is there. So many of the organisms that are present around these vent fields are unknown to science, unknown to mankind. And so every time this team and their other colleagues in the broader community, every time biologists like that have an opportunity to investigate these environments, they continue to find new species, new animals that are unknown to them. And even as they're investigating animals that have been seen before, they are learning fundamentally new things about them. You know, for instance, from the 2018 work, Shauna Gofrady and her colleagues showed that certain scale worms are also chemosynthetic, something that was not known and perhaps not even really suspected before. So the very fundamental explorational science is happening on that leg. Not only at the macro scale, but also the microbiological scale, which Victoria and her team, John Magyar and their students, will be doing sediment sampling and looking at the microbes, trying to grow the microbes and identify the microbes that are in those sediments. And in many cases, just the very basic mix of what's growing in there is unknown. So very first order information. And then even if you understand what's there, you don't necessarily understand what it's doing. What role does it have in the ecosystem? What are they eating? What's eating them? These are all things that are unknown at every scale, from the most lovely large animals down into the microbes. There's more mystery here than there is knowledge at this point. And so this broad interdisciplinary team of biologists are looking across all the things that live in these places. They're looking at them individually, and they're trying to understand how they integrate together into this whole amazing ecosystem. So, hope that at least partially answered your question, but I would encourage you all to follow the dives that happen next week and the week after, because it's going to be really, really cool.
Yeah. They're looking forward, actually.
Well, just to prove that I'm tough to shut up, Jeremy said, I guess how species get here is also interesting, and actually that is another aspect of this that this team, particularly Victoria and Shauna, have had a lot to do with the very basics of trying to understand the processes of the dispersal of different species to different vent habitats. And so there's real questions about why are the vent species that inhabit some vents different from other vents that are geographically pretty close, and why are they in many respects the same? You know, what controls those differences? And the thing is that the majority of these species propagate through larvae. So they produce larvae that go up into the water, and only the larvae that find a new appropriate habitat will survive. So the movement of animals from one vent habitat to another depends on transport through the water, through the ocean. And the complexity of that, the interplay of that, and the evolutionary history of that is really interesting and not well understood. And that comes back to some of the work that we're doing in studying the regional scale morphology and formation of these basins. That is, the rifting processes that have produced these basins in which seafloor spreading is starting has a history, and the existence of these vent habitats has depended on that rifting history, that geologic history. So how long vent habitats have existed in this part of the world is dependent on the geologic history. Where they are depends on the geologic context right now. We know that there are vents in Guaymas Basin up to the north. We know that there are vents in the Alarcon Basin on the hard rock spreading center of the Alarcon Rise to the south. We know that there are some vents on the transform faults in between South Pescadero and Alarcon Rise, and also on transform faults up near Guaymas. In between, in the Carmen Basin, the Farallon Basin, the North Pescadero Basin, we really don't know where vent fields exist because we haven't adequately explored those places. And so that aspect of that question of how do the animals get to these vent sites, and what's the evolutionary history, and what controls which species are where, our ability to investigate that right now is very limited by the fact that we don't know where all the vents are. And so we can't really start to answer those questions satisfactorily. So the large-scale mapping and exploration is an important part of the overall scientific endeavor, that's important even down, in terms of relevance, down to people like Victoria that are doing push cores and looking at what microbes inhabit the mud. So that's why it's so important that we're able to work together in this broad interdisciplinary team. So I think we are well underway in what is going to prove to be the last heat flow measurement. Is that correct, Florian?
Yes.
And it looks like a really good one. What was the last heat flow measurement, Florian?
7.25.
7.5. So, the latter part of today, as we worked up the east side of this vent field, east of the chain of large edifices, the ground has been really hot, and there is a lot of conductive heat transport out of the seafloor here, and the signal shows quite a bit of variability. It's really very interesting, and we know that it reflects the structure that's underneath us, that controls the flow of the hydrothermal fluids. But we're a long ways from, as we sit here now and we're making these measurements, we're a long way from making sense of it. We've got a lot of information, but it's going to take the team a great deal of work, including ultimately doing some numerical modeling to try and understand what these heat flow observations are telling us about the processes of fluid flow in the subsurface that results in the existence of these vents.
Thank you, Dave. That was great. You're natural to do that.
So if you follow the ocean crest out, yeah, it becomes hard to direct. What it is pockets of hydrothermal deposits. In this case, hard rocks, right? But when it gets to hard rock, there's still a lot of sediment underneath. Calling it calcium sulfide deposits. These carbonates, well, the carbonates dissolve, but once they're buried by sediment, they won't. But up to that point, they'll dissolve because it's deep, it's below the surface. But what happens with the sulfides is they start out kind of hard and they make these chipples and stuff. But those minerals weather really fast, and so in a lot of cases, in just a few years or a few tens of years, they go to clay minerals that fall apart and they just become this pocket of clay, and then you get pelagic sediment. But places where you built up substantial deposits like that, that's where you get copper ores, right? Like cypress or whatever. So typically great. See, we'll correct the things that are wrong, but a large proportion of the commercially viable copper deposits around the world originated on the seafloor of ocean and spreading centers as long as
Okay, guys. Well, we've waited for the last measurement of the day to be done. I'm just gonna address some of the questions on the chat. So the microbial ecology and the biology here have been investigated by the same team that is going to be on board next week. So this is a follow-up of their work, and I would even say that they are the experts of this area and have discovered many new species and characterized them. So yes, it has been studied in the past, but by the same group led by Dr. Victoria Orphan from Caltech. So if you are anti-biology, then next will be like a dream, because they will be doing a dive every day that they're here, if everything goes okay, and so you will have the chance to look at the animals and all the ecology very close, and see them taking samples and of the animals and sometimes the water as well. And there was somebody asking what's coming up from the vents in terms of chemistry. Well, that's one of the reasons we are taking samples, because each vent has a particular chemistry, and so it's part of this.
Ready? Get the probe up, please.
Probe's coming out. Thank you.
Okay. Yeah, I told you, 2 minutes. All right, guys.
Awesome job.
Well, thank you so much, guys. This has been a just about perfect day
Probe's
for
out.
us.
Thank you.
We're done, Mr. Gerald. Right on time.
Nice. I was just about to ask you. If we stayed out longer, that's okay. Just gonna affect the bottom time of the next time.
No, that's exactly what we didn't want to mess with it.
Yeah.
So,
We do have one dive early in the morning tomorrow.
yeah, I'm gonna give you actually the floor to finish things off and just let the audience know. Okay, guys, so I'm gonna leave you with Dr. Carres, who's gonna explain what's gonna happen tomorrow, which is gonna be the last dive for our expedition, and so it's gonna be a particularly different dive from what we've been doing for the last week, and so signing off here in the name of on behalf of my heat flow team, we thank you for your questions and your continuous presence and your interest in our science. Okay, see you soon, guys. Bye-bye.
All right, well, this is Dave Caress again, as they are stowing the pokey thing and preparing to come off bottom and ascend up to the surface, which will take us, as it always does from this depth, about 2 hours and 15 minutes or so. So this is the end of the dives in South Pescadero Basin for leg 2 of this expedition for interdisciplinary study of the hydrothermal vent fields in South Pescadero Basin. It is not, however, the very last ROV dive of the expedition. What's going to happen here on the ship is that the ROV will be on deck between 6 p.m. and 6:30 and so forth. And after that, the Falkor is going to start transiting back to La Paz, Baja California's south in Mexico. Early in the morning, we're going on our way back to La Paz. We are going to stop and do an ROV dive that's relatively shallow in order to obtain some rock samples that are relevant to the history of rifting here in the Gulf of California. That is going to, we expect to have the ROV going into the water at around 5 a.m. our time or a little later, and that's equivalent to U.S. Central Time. So 5 a.m. La Paz time. And we're only going to 700 meters to start the on-bottom part of that dive, and then we will be working our way up a sharp scarp, an east-facing scarp, to about 300 meters or so. So we'll probably be on bottom somewhere between 5:30 and 5:45 in the morning, and we're only going to be on bottom for about 2.5 or 3 hours. Again, the purpose of that dive will be to obtain some rock samples. We're hoping to find some outcrops along this scarp that can be sampled, and then hopefully these rocks will contain information both in terms of their composition, what kind of rock they are, how old they are, but also in evidence for deformation that might be related to the faulting that occurred along this margin. So we'll be trying to get those rock samples. This is to support the research of Ron Spells Madero, who was the chief scientist of leg 1 of this expedition. Support the research of Ron and his students, who are working at the history of the rifting and the mechanics of the rifting here in the Gulf of California. So we have one more dive in this expedition, but you gotta, at least if you're in North America, you've got to get up pretty early to catch it. And of course, as has been mentioned many times, then the ship will go into La Paz. We will change science parties. I will be among the Falkor, and that new team will be back in the South Pescadero Basin, doing 11 to 12 ROV dives, depending exactly on how the logistics works out. So I'm not going to sign off of the expedition as a whole. I'll be doing that at something like 10 o'clock tomorrow morning. But this is signing off from the South Pescadero Basin for leg 2. And for those who won't join us again in the morning, because it's way early. Thank you so much for paying attention. It's for oceanographers. It's a novel experience to be here in a shipboard context where we can share what we're doing more broadly, and it's as one develops an understanding of how to integrate that in with everything else we're doing. It's pretty fun. So thank you so much for your questions and paying attention. And hope to be in this chair again sometime in the not distant future. Bye.
Bridge, RV.